السلام عليكم جميعا. Welcome to the second day of your basic neuroscience lecture. اليوم راح نبني على المفاهيم اللي تكلمنا عنها بالمحاضرة الأولى وراح نحاول we go from single neurons to how this single neuron can encode an environment and then how the brain reacts to this environment. خلينا نقول okay? Alright, so as an outline في البداية راح نتكلم عن what is the neural code. بعدها بنتكلم عن sensory systems and how they can encode the environment efficiently وكيف this sensory information is used to make decisions. After that, we're gonna briefly talk about reward and learning وراح ناخذ راح نتكلم عن reinforcement learning specifically. And in the end, we're going to talk about what happens when these systems go wrong. بناخذ Parkinson's as an example, and we're going to delve into how dopaminergic circuits are affected in Parkinson's. بس حابة أقول إن the concepts اللي we're going to cover during this lecture حتكون شوية يعني سميكة فلا don't stress yourself إذا حسيت إنه it's too much لأنه every part of this ممكن نقعد نتكلم عنه 3-4 lectures at a time. المبدأ هو إنه أنا بس أبغاكم تفهموا in general إنه نوصل من a single neuron ل how we navigate this environment and how the brain really makes sense of how the brain really makes sense of all the information it's getting. Okay. So إحنا في اللكشر اللي قبلها تكلمنا عن الخلية العصبية و how the activity of this neuron يتحول إلى information and builds core circuit motifs. So we briefly talked about like how excitatory neurons and inhibitory neurons connect to each other. From this core circuit motifs, usually there's a large-scale architecture plan, such as continuous topographic mapping that is kind of depicted here, that enables specialized functions of the brain. So integrating the structure and function of neural circuits is kind of a central goal of neuroscience, and investigating their development and their evolution is also crucial, provides crucial insights. الأيديا هو إنه مثلا الإكزامبل اللي معطينها هنا إنه مثلا a neuron gives you the information in the form of a letter, the circuit motif يعطيك إياها in the form of a word. بعدها عندك large scale architectural plans that connect different brain regions together. ممكن these different brain regions now form a sentence, and then the brain can write an entire article of the things that it's seeing and how it's reacting to how it should be reacting to it. Okay. Right, so from our most basic understanding, David Marr proposed that cognitive processes have 3 levels of description. So any information processing system can be analyzed at 3 levels. The first one is like a computational problem that the system is trying to solve. The second one would be an algorithm that the system uses to solve that problem. And 3, how that algorithm is implemented in like the physical hardware of the system, or let's say مثلا the neurons and the circuit motifs that are built and the different brain regions. Okay. So in the past 20 years, neuroscience research has shifted from area-wide manipulations زي ما تكلمنا باللكشر اللي فات عن Phineas Gage وكيف we know how different brain regions work because we have very basic early studies that do lesions and remove whole brain regions, and so we know in general what they do. So these are area-wide manipulations such as lesions or pharmacological silencing. But now we do more circuit-level things that enable researchers to target and define subsets of neurons, and that's what we were talking about in the other lecture اللي هو optogenetics و circuit manipulations من هالناحية يعني. For many decades, researchers مثلا use electrodes to monitor the extracellular activity of single neurons, and the activity pattern of each neuron was often characterized by tuning curves or receptive fields اللي حنتكلم عليهم بالsensory systems, بالsensory processing والsensory systems اللي we're going to take as an example in this lecture. Newly developed tools that can record thousands of neurons simultaneously, enabling the study of population level dynamics. وإحنا هذا اللي إحنا نبغى نركز عليه، نبغى نركز إننا من single neuron للpopulation level, what computations are being done by the brain that allow us to navigate our environment and maximize survival. Okay, so traditional laboratory experiments in artificial settings, مثلا the water maze or lever pressing, often restrict animals to unnatural behaviors or stereotypical movements. But الحين neuroscientists are trying to study more naturalistic behaviors, مثل hunting and hunting to understand how the brain does things in more realistic settings. Okay. So خلينا ندخل الحين في how do we study what is the neural code, okay? So Pitts were the first to argue that neural activity is computation, and that neural computation explains cognition, okay? So as you can see here, this is called a raster plot, and it's basically the activity of neurons throughout a behavioral paradigm. You can see إنه مثلا some neurons respond, some don't. Each neuron is responding to something special in the environment. So you can see there's a heat map where the neuron is responding either to the stimulus or the action or the outcome of the action after the action is taken, okay? So what we're going to try to attempt through this lecture is understand how do we think about this activity? Is a neuron signal a message, or the population as a whole responding and creating a complex code? So ultimately, we must use the information in the environment to take appropriate actions to maximize our survival. Okay. One of the ways that we typically think about neurons, either as encoding. So encoding is how does a stimulus cause a pattern of response? And because not neural systems are noisy and contain all kinds of relevant and irrelevant information, we think of encoding and decoding in a probabilistic way. So here I'm showing you the probability of a response given a stimulus, which is conditional. وإحنا بالنهاية we want to understand what is the relationship between stimulus and response, and attempt to predict either the neural activity, and here they're attempting to predict the neural activity from the auditory representation by finding a set of weights for each feature that minimizes the difference between the true and the predicted response. Okay, whereas decoding is the opposite. What does response tell us about the stimulus? How can we reconstruct what the brain is doing? This is often done in controlling neural prosthetics. We think of it again as a probability of a stimulus being shown given the response that we recorded. So in decoding, brain activity is recorded by electrodes and is used to make discrete predictions of the category of this stimulus. Okay, so we're going to take the visual system as a kind of example of what encoding a response can look like. So at its core, vision is about detecting light. And you probably heard of rods and cones. These are photoreceptor cells in your eyes that physically detect photons of light. Photoreceptors are distributed across our retina. Each cell can only detect light coming from a particular point in space. To perceive an image, our brain has to take this information from all these tiny photoreceptors and clump it together through the optic nerve and send this information to the rest of the brain. The information goes through the thalamus. From the thalamus, it goes to higher order areas, within the visual cortex, where it's taken, where the processing and creating an image of what you see happens. Okay, so how do we go from detecting a photon of light to representing the entire environment or seeing? So cells that send information from your eyes to your brain display fairly simple responses to light. So each cell fires when you shine light in a specific small circular area of the visual field, with different cells responding to light in different places. So here you can see the response of a cell to a circular spot of light. And this is from the seminal work of Hubble and Weasel. These neurons respond this way because they get positive input from the receptors that are clustered together in a specific area of the retina. These retinal output neurons are inhibited when you shine light just outside of their preferred circular area. So they're called centers. This is why they're called center surround cells. This suppression is due to the inhibitory input from the corresponding photoreceptors. So basically, these center surround cells are really good at detecting light, little circles of light. This is useful, but it's a long way from recognizing what is happening in the real world. Okay. So, like I said, you go from the retina to the thalamus, and the thalamus relays the information of center surround cells to the visual cortex, a higher order brain center. So, Hubble and Weasel discovered that there were things that this is where things really got interesting. So, their basic approach was to record activity of neurons in cat brains while presenting various visual stimuli, patterns of light. They did this a lot, and they found that actually they had very little success. So no matter the size or position of the circle, the neurons in the visual in V1 were not responding to any of this. And they just couldn't care less. So it turns out that actually these cells were responding to the edges of the slide. And that's when Hubble and Weasel discovered that there are cells actually specialized in detecting lines, which is a basic feature of the visual world. So they recorded from that neuron for 9 straight hours, and they called it the simple cell. And they have this that each center, and they found that each center surround cell in the thalamus is activated by a dot of light in a particular location. But what do you get when you put a bunch of dots side by side? You get a line. And so Hubble and Weasel hypothesized that each simple cell might get a positive input from a bunch of thalamic center surround cells, and you can see that in the description in the image here. So center surrounds who preferred areas are arranged in a straight line. So these simple cells only fire if enough of these inputs are simultaneously active, which occurs when you shine the light in a straight line in the correct orientation. A larger shape, like a square, wouldn't work for center surround cells. Okay, so this is essentially what neural circuits do. They transform basic signals into useful, often sophisticated information that we can use to understand and interact with the outside world. Okay. One of the more recent papers that came out actually showed that cells in the hippocampus responded to, there was large spikes of activity, and these cells actually responded to, impressively, Jen Aniston and Brad, pictures of Jennifer Aniston and Brad Pitt, but not Jennifer Aniston by herself specifically. So these neurons are specific, and you can think of them as embodying a concept. So now we've gone from basic lines to a representation of a person, a representation of a couple. Okay, so brain regions increasingly build a more complex picture of the world and represent the stimulus, starting from simple shapes from the retina and the LGN, to V1 where you have these oriented edges, to V4 where you have contours and edges, to higher up in the brain where semantic representations take form, e.g. a person or two people together. Okay, so we're going from geometric features to more semantic representations of the brain. So you have this kind of assembly of features from simple to complex, which is very successful in machine vision models. So these regions are also massively connected. So although the thalamus is thought to be like a relay station for information coming in from the retina, it also receives a lot of feedback from areas that it projects to, suggesting that these representation feedback to control how information is represented in your brain. So here we have a role for learning and expectation. What you think you're looking at can shape what you see. And this is an example of an artist, Louis Wayne, who really liked drawing cats. And his drawing of a cat actually changed and his style changed as his psychological condition deteriorated. So Louis Wayne was thought to have schizophrenia. And as his schizophrenia got worse, the image of the cat, as you can see, got more and more abstract, and even you could say maybe more and more menacing. Okay, so we now know that we now can see how sensory systems build a representation of the world, and this is using simple we're taking the reaction of a photoreceptor to a photon of light, and we're really building an entire world around it. So this input is also taken to make decisions and navigate the environment. And you can think of it in this kind of way. So you accumulate sensory evidence. There's some kind of computation that happens in the middle. After this computation, there's a decision being made and an action being made. And then the next couple of slides, we're going to talk about after completing the sensory evidence, what kind of model systems are able to put things together so that you navigate your environment correctly. Okay, so how does the brain do this? So one way the brain can kind of learn from what you're looking at is by statistically approximating what we see by what we know. So خلينا نتخيل انك كنت تمشي في الشارع وشفت you saw like a brown fuzzy shadow running, okay? ممكن يكون هذه قطوة الجيران ممكن انه يكون كلب بس مخك ما استوعب there was never not enough sensory evidence for you to know. So one of the ways the brain can compensate for this is by what we call Bayesian statistics, Bayesian inference, okay? So it's likely that you're going to decide what you saw by using both your expectation of either the animal showing up at your garden, like the neighbor's cat, or the visual features that you glimpsed that just caught your attention. So this is an example showing that humans utilize the limited knowledge about the world and the uncertain information about the environment to make decisions in a process involving probabilistically inferring hidden causes from these observable sensory inputs. So Bayesian decision theory specifically specifies statistically optimal solutions for any given inference problem. And it's kind of a good benchmark model for understanding probabilistic computations that take place within the brain. So the Bayesian brain considers the brain as a statistical organ of hierarchical inference. So احنا مو قلنا we went from a spot of light to a line to more complex representations of the environment. These can help predict current and future events on the basis of past experience. So according to this theory, the mind makes sense of the world by assigning probabilities to the hypotheses that best explain and usually are uncertain sensory data, and then continuously updating these hypotheses according to standard probabilistic rules of inference. So Bayesian decision theory is what Marr would consider a computational level analysis. It concerns the goal of an information processing system and why is this goal appropriate. So there are two parts of Bayesian decision theory: a generative model to specify a probability of an observed result from hidden states, the hidden states being something you're unsure of, a gain of function that specifies the gain of taking a particular action for a particular situation. And the two components of this general model include a belief about how likely the state is to occur before receiving an observational prior. The prior is what you've learned before. And the probability of this observation conditional to hidden states and the likelihood that it would happen. So following Bayes' rule, by multiplying the prior by the likelihood and normalizing it, optimizes the information about the hidden states probabilities given that observation. So brain regions within a neural system are hierarchically organized, and each level of hierarchy sends a top-down signal to predict inputs of lower levels. Okay, so Bayes' rule helps you essentially create an estimation of your environment from the things that you learned to help you make decisions and pick the appropriate response. Okay, so one of the ways you can go on by so one of the ways in which the brain also predicts mistakes in these kind of mistakes in the reactions or the things that you expect or the environment that you expect or the response that you expect is through something we call a prediction error that helps you adjust your expectations. Okay. So this is an experiment in which a thirsty monkey is between two levers. The monkey has been trained to perform a simple choice task. And during the early phases of this process, the monkey behaves somewhat erratically and the neurons are silent when the start cue is presented, but responds strongly whenever it receives a reward. So as the monkey continues to perform the task, however, both the behavior and the activity of these neurons changes. And the monkey becomes focused on his lever pressing and the lever that leads a reward. And this happens, the response of the neurons to the juice reward dies out. However, the interesting thing that happens is that when the monkey is expecting a reward and it doesn't get it, the reward is emitted, the animal is experiencing something called a negative prediction error. These monkeys were first trained to anticipate a reward with a visual cue, but on rare trials, they simply didn't get the reward. Under these conditions, they found that the neurons were responding to this kind of emitted reward with a reduction in their firing rate from the baseline levels. So reward prediction error responds at the time of reward, which you can see on the right, and the reward prediction visual stimulus in the middle. And the dopamine, dopamine neurons are activated by the unpredicted reward, eliciting a kind of positive reward prediction error. Whereas some cells show no response to the fully predicted reward, so there's no prediction error, which is zero error shown here. And then it is also depressed by the emission of the predicted reward, eliciting a negative prediction error. So here you can see dopamine neurons are responding to the expectation of the reward. Whether it's there or not, these neurons are calculating what happens when we don't get a reward. Okay, so the main idea here is that your brain is responding to the outcome of your prediction. In this term, it's responding to the outcome of whether or not it got a reward that it predicted. Okay, so the way dopamine responds to reward is the basis of something called reinforcement learning. So as I said, that an important problem that faces decision making is learning and usually learning happens by trial and error. Which decision to make and when to obtain reward or to avoid punishment. In computer science, this problem is known as reinforcement learning. And one of the most fundamental goals of any human is to make the accurate prediction of future events in order to be prepared when this expected future arrives and adapt your behavior accordingly. So this process is generally involves an improvement of these predictions. So dopamine here functions as a reward prediction error signal. And put simply, dopamine calculates the difference between the reward that was expected and then the reward that was actually received or not received. So The data currently suggests that phasic activity in the midbrain and dopamine neuron encodes this reward prediction error to guide learning through the frontal cortex and basal ganglia. This activity is now believed to signal that a subject's estimate of the value of the current future event is in error, and it indicates also the magnitude of this error. So this is a kind of combined signal that adjusts synaptic strength in a qualitative matter until the subject's estimation of the value of the current and the future event is accurately encoded in the frontal cortex and the basal ganglia. So altogether, this biological activity is encoded and creates this kind of reinforcement learning algorithm that has achieved a lot of success. It's good to kind of remember here Marr's levels of analysis, and reinforcement learning is another way that the brain takes a computational problem and leads to a host of algorithmic solutions that seem to be implemented in the human and animal brains. Okay. So in summary, well, for the summary for this section is that sensory systems can encode the environment efficiently, and even if we don't know something about the environment or something that we're uncertain about, or we have half the information about, Bayesian brains are good at predicting that. Neurons can respond to simple and complex stimuli. This activity builds a representation of the environment. This representation that we build is used to learn and predict the environment and help us maximize survival and maximize reward. Now, in the second section of this presentation, or this lecture, we're going to go through, as I said, the dopaminergic circuit to kind of understand and put everything together. Okay, so let's take a bit of a deeper dive into dopamine and dissect the dopaminergic system. So dopamine, as I said, plays an important role in neuromodulation, such as motor control, motivation, reward, cognition, cognitive function, maternal and reproductive behavior. Dopamine receptors are expressed throughout the central system. Each dopamine receptor is thought to take on a specific role within the system. So مثلا D2, locomotion, attention, sleep, memory, and learning. D3 receptors are cognition, impulse control, attention, and sleep. D5 are involved in decision-making, cognitive control, and attention. Okay, so as a major neurotransmitter, also dopamine regulates motor neurons, and all these kinds of a diverse, really, array of behaviors. Physiologically, dopamine cannot be simply classified as an excitatory or inhibitory neurotransmitter, since it can bind to different G-coupled protein receptors and is differentially modulates adenylate cyclates depending on the type of dopamine receptor that is involved. So in schizophrenia, a prolonged decrease in the prefrontal cortical activity is proposed to reduce this dopaminic release, and over time, compensations in the system increases the overall dopamine responsivity and thereby causes increased phasic dopamine release that causes abnormalities in the way the environment is perceived. So we said there's transient or phasic dopamine release that's caused by dopamine neuron firing. And then we have a sustained background tonic dopamine release that is regulated by prefrontal cortex afferents. Okay, so there are four main dopaminergic pathways. This is the mesolimbic pathway, the mesocortical pathway, the nigrostriatal pathway, and the tuberoinfundibular pathway. Midbrain dopaminergic neurons from the ventral tegmental area, the VTA, project to the prefrontal cortex via the mesocortical pathway and to the nucleus accumbens via the mesolimbic pathway. So the VTA is also the region of dopaminergic projections to the amygdala, hippocampus, cingulus gyrus, and olfactory bulb. Electrophysiological studies, which localize on brain stimulations, have implicated the mesolimbic dopaminergic pathway in positive reward and appetitive motivated behavior. Overall, dysfunction in these different pathways is implicated in a wide range of processes that result in disease or disorders that are ranging from addiction to movement disorders such as Parkinson's. So we'll talk about Parkinson's a little bit. Parkinson's disease is the most common neurodegenerative movement disorder, and it's pathologically caused by the loss of these dopamine-producing neurons in the substantia nigra comparta, and the presence of intraneural aggregates that are called Lewy bodies that are enriched in alpha-synuclein. So the disease presents as a cluster of motor symptoms called Parkinsian symptoms, which is like bradykinesia, tremor, rigidity, and instability. Okay, these are common attributes to the loss of striatal dopamine, as well as there are non-motor features that include depression and cognitive impairment that are thought to be a result of the loss of dopamine and the degeneration of circuits that are that project to the amygdala or the hippocampus, مثلا. And the number of dopaminergic neurons in the substantia nigra decrease at around a rate of 4% per decade as part of the normal aging process. However, when you have Parkinson's, the decrease is around 70% of the expected in age-matched controls. So the classical model of how Parkinson's arises from the loss of striatal dopamine content is based on the functional connections within the basal ganglia. So a decrease in dopamine level reduces the activity of the direct pathway and increases the activity of the indirect pathway. So this schematic represents the normal nigrostriatal pathway in red, and it is composed of dopaminergic neurons whose cell bodies are located in the substantia nigra comparta. These neurons project to the basal ganglia and synapse in the striatum. And we're showing you here the normal pigmentation in the SNP, which is the substantia nigra, produced by neuromelanin within the dopaminergic neurons. So this schematic represents the disease nigrostriatal pathway in red. In Parkinson's disease, the nigrostriatal pathway degenerates. And this is a marked loss of dopaminergic neurons that projects to the putamen, and a modest loss of the projection to the caudate. So the photograph shows you the depigmentation and the marked loss of dopaminergic neurons. Immunohistochemically, if you look at the labeling of the interneural inclusions, these are termed Lewy bodies. And the immunostaining within shows that alpha-synuclein reveals the immunoreactivity and the increase in these kind of aggregated Lewy bodies that cause degeneration of these cells. Okay, so we talked a little bit about how briefly about Lewy bodies and alpha-synuclein, and now I'm just gonna go through briefly what happens. So currently it's suggested that the accumulation of these misfolded proteins is likely a key event in Parkinson's neurodegenerations. So there's pathogenetic mutations that directly induce abnormal protein conformations, as believed with the protein alpha-synuclein, or damage to the ability of the cell to actually degrade misfolded proteins, such as damage in Parkin or UCHL1. Oxidative damage linked to mitochondrial function is also involved. An abnormal dopamine metabolism promotes the misfolded protein conformations, but it remains unclear whether this misfolded protein directly cause toxicity or damage the cell via the formation of protein aggregates called Lewy bodies. So there's a controversy that exists whether Lewy bodies promote toxicity or protect cells from harmful effect of misfolded proteins by sequestering them in insoluble compartments away from cellular or elements, because there's an issue in lysosomal degradation. And there's also an issue with oxidative stress and the activation of programmed cell death machinery that's believed to be factors that trigger the dopaminergic neurons' death in Parkinson's disease. The loss of these neurons also affects the activity of these neurons, and you can see here a comparison between healthy cells in various compartments of the VTA. So you have you can see that some of these cells are either there's either an increase in activity, which is probably a compensatory mechanism, or a decrease in activity due to degeneration. Okay, so we talked about how dopamine causes a loss in motor function because of the loss of the nigrostriatal pathway, but also there is a plethora actually of non-motor symptoms that include hyposomnia, sleep disorders, depression, and they appear in a spectrum of patients with PD, including individuals that have a genetic cause to PD. One of the ways in which we treat Parkinson's is deep brain stimulation. So deep brain stimulation works as a pulse generator. So it's like a device, like a heart pacemaker, is placed under the skin and the chest, or around the chest or stomach area. It's connected to 1 or 2 fine wires that are inserted in specific areas of the brain where there is dopaminergic neuron loss. And this pulse generator is switched on. Electrodes deliver high-frequency stimulation to the targeted area. This stimulation changes some electrical signals in the brain and reduces the motor symptoms of Parkinson's. Okay. Usually they're implanted in the subthalamic nucleus, and DBS is known to improve the motor functions for up to 10 years, although the magnitude of this improvement tends to decrease over time. So basically here you have a loss of these neurons, and we have a compensatory effect to the loss of these neurons within Parkinson's patients that allows them to live a somewhat normal life. Okay, thank you for listening. I hope that wasn't too overwhelming. I know we covered a lot of concepts within this within a short time, and we went over them super briefly. But the idea here is that you get a general understanding of how we can go from single neurons to circuit to complex computations in the brain that allow us to navigate our environment and behave appropriately. Now, I look forward to seeing you in the Q&A. I'm sure you will have a lot of questions, and thank you again.